Okay, good morning, class. How are you today? So, welcome back again to our discussion in InBt121 lecture. I hope you have submitted your reaction papers on cell immobilization, is what I have given to you last week. Let us now continue with our discussion in lesson 2. Okay, the lesson 2, it is entitled Characteristics of Immobilized Cells. So I will not dwell with this one to pre-test because I have already given it to you in the VSUEE. So I encourage you to, and also I have already, we have already answered this question. So just have to refresh your mind about it. So it's on the handout. So please study for the for the incoming final exam on December. Okay, so malapit na tayong matapos class. Okay, so for the review, the lesson 1, so you have to recall the different types of immobilization techniques and give examples of each type. So I will require all of you to submit the answers that will be provided in the BSUEE, even though it's not graded, because this is one way of the of refreshing your minds about the lesson class, okay, and how it works, the mechanism of it. So this is the diagram or the illustration of the different methods of immobilization. For the outcomes of this lesson, you will be able to explain the different characteristics of immobilized cells in terms of function environment and product formation. And second, or the last outcome or the objective is to identify examples of immobilized cells used for the production of important compounds. Okay, so we have already known, class, from the previous lesson or the lesson 1 that um, there are many types of methods of immobilization that would um, give an advantage to uh, the fermentation or to the production, um, overall production, that it may reduce labor cost, and it may um, increase the concentration of the product of the biological product that is really wanted to be produced. Yeah. So, but the question is, have you ever wondered why? What happens when the cells are immobilized? Why is it that they they still can produce the kind of product that they have to produce, or the metabolites they have to, and why is it that they can still grow and they can still consume the nutrients of the fermentation media, or the media itself. Yeah. So this is the question: what happens when the cells are immobilized? Now there are many factors that the cells are will grow in a particular medium. Okay, so there are many factors, there are many conditions that has to be considered in order, di ba, when you took the microbiology course. There are many factors that affects the condition of the bacteria, and iba din sa yeast. It's different also in the for the molds. Okay, so for the bacteria and molds, yeast, ganun. So there are many differences, and also these conditions are really critical in order that the metabolism of the microorganisms will continue to function, right? So that is why there are many studies that has been conducted in order to optimize to determine in what condition that this bacteria will grow uh, at its highest concentration of biomass. And also there are also studies that identifies the um, identify the optimum condition which the biological product that you wanted to be produced is at higher concentration. Okay, so now Microbial cells have behave according to their environment, right? So when it is immobilized, the microenvironment of microbial cells is relative to the immobilization method used. Now, the altered chemical composition and the physical interaction between the matrix material and the cell has a significant effect on the cell's function. So imagine, class, there is the interaction between the physical interact between the matrix material and also the cell wall, right? So this lesson will discuss the effect of immobilization upon the cell's physiology for bacteria and fungi. Okay. So this is the summary of the observed effects of fungal cell immobilization in its physiology. Okay, so here is the immobilization effect on the first column. Then you have the microorganisms used and then the immobilization system. As you observe, class, that there are many immobilization effects that are written here, and like the altered osmotic conditions in microenvironments, like the exchange of nutrients, the cell to cell, the exchange of nutrients for the entrance of nutrients into the cell, right? Diba? Decreased ethanol tolerance, there is also decreased ethanol tolerance, and there is also an effect that it will increase ethanol tolerance, meaning hindi siya mamamatay ang bacteria like the Saccharomyces cerevisiae when it is immobilized. So, hindi siya mababahala na the biomass of the microorganisms will reduce because of the presence of alcohol. When particular, it is in the wine fermentation, the ethanol fermentation, and using the immobilization system. Iba din yung effect ng paisha, which is paisha is also a microorganism for the ethanol fermentation. So it decreased ethanol tolerance. So there will be a possibility that when the ethanol concentration will increase, so the K carrageenan method for Paisha stipitis immobilization will not be effective for the reused because of course there is a possibility that it is now susceptible to ethanol, diba? So mamamatay yung mga biomass because of this observed effect because of the immobilization. So that is it. So there is also an increased organic solvent tolerance and also decreased inhibition of cell growth and metabolism by better carbon dioxide removal. So, and many more. There are many observed effects in terms of increased plasmid stability, in terms of protective microenvironment, influence of mass transport limitation, and also change of productivity, and then change morphology and composition. So there are many, and also there is also an enhancement of enzyme stability. So later I will also give the detailed journal article in which you can really read. You can have a bit better perspective and wide understanding on how to uh, the different effects, diba? Different effects of the immobilization differs in the microorganisms and also differ in the immobilization used with that particular microorganism. So that's it. I will not dwell in that much more. So yeah, we'll just have a summary of the effects or the different effects of the immobilization in the bacterial cell. Well, in terms of plasmid stability, class, are you familiar with plasmid? Ano nga ba ang plasmid, class? Okay? So, think of it. So, ano ba yung plasmid? Ano nga ba yung plasmid, di ba? Plasmid is where is it located? Where it can be found? It can be found in the bacterial cell. So there is no plasmid in the eukaryotic cell, diba? So the plasmid is only for the bacteria. So the plasmid, it is a circular what? Circular DNA. It contains DNA. It is a circular DNA. That's why it is called extra chromosomal DNA. So meron tayong plasmid, meron tayong genomic DNA in which in the bacteria it is naked, diba? So unlike with the eukaryotic cell that it is packed in the nucleus and then also it's packed with the histone proteins. So marami. So please review your molecular biology. So you can have that. And while um, unlike with the bacteria class that uh, the genomic DNA is in naked form, meaning wala siyang, it's not packed on histone and also it's just in the cytoplasm. Yeah, so it's, di ba, ang eukaryotic cell is compartmentalized siya. Merong vacuole, merong chloroplast, ganon. And the DNA is located or is located in the nucleus, but there in the bacteria there is no, it is naked. But yeah, so much for that. But then this plasmid is the extra chromosomal DNA in which this plasmid contains information about the plasmid's antibacterial ability or the antibacterial effect against the against antibiotics, and also it also confers many peptides that may form a defense mechanism against the other bacteria, right? So there are many, and that is why plasmid has been used as an advantage for gene cloning, because plasmid can be transferred to another using many different methods of transformation. Later in InBt121, you will learn all of these things, the different methods of transformation. So in the plasmid, you can manipulate the plasmid, but then they're talking here about the plasmid stability of the bacterial cells, in which it is also has a significant in the cells metabolism or physiology. Like for the bacterial cells, like for example, in the recombinant bacterial cells, right? When you say recombinant bacterial cells class, ano ba nga ba yun? So when you say there is a foreign gene inside your plasmid, right? So meaning the plasmid is cloned, meaning sa BT134 yun siya class, meaning you have the gene of interest from the other bacteria, like you wanted to express this. For example, insulin, right? Diba ang insulin, the DNA of insulin is also, it can be found sa humans, sa animals. Now, this insulin, because of the many increasing demand for the insulin protein, because of the increasing patients of um, of diabetes, yeah, increasing diabetic persons. So there is a higher demand of insulin. That is why they opt for the recombination of the insulin gene and then cut it out from the, actually cutting it out, extracting its DNA and then cutting it off using a restriction in the nuclease. And then you have to transfer this in a plasmid, and then you paste it, copy, paste, tapos ganon. DNA ligase, yung magpipaste, actually, it's not, hindi lang siya parang ano lang dito sa computer class, but then it is a DNA ligase that will act on the pasting of the gene of interest to the new, the plasmid, and then also this plasmid will then be transported or transformed into E. coli. So, minang E. coli ang host organism. Minang ang E. coli ang saman, ang E. coli ang mo-express sa insulin na protein. That is why it is called recombinant bacterial cells, because ang insulin kay dili man siya naturally produced by the E. coli. So, recombinant bacterial cells, meaning importante ang plasmid nga naa diha, because the plasmid will be contains the foreign gene, di ba? It contains the foreign gene that is your target product, okay? The stability of the plasmid containing the foreign gene can be increased upon immobilization because um, there are factors in the heart, there are factors outside the environment, diba, that would decrease the stability of the plasmid and it is prone to mutation. So, kana siya. So, meaning there is at risk na kung there is a uh, thinner barrier between the cells and the environment, meaning there is a possibility of mutation of the plasmid, and then the uh, the bacteria is really fast in acquiring many different in many mutations. So that is why when the recombinant bacterial cells are immobilized, so there is a increased stability of the plasmid, meaning it is not prone to mutation. It is not prone to meaning the plasmid can be still replicated to another Ayan, can still be replicated with another during the cell division. But yeah, that's how it is described here, no, because of the thicker barrier of the matrix material and the cell. Okay. How about with the fungal cells? They improve plasmid stability in immobilized uh, Saccharomyces cerevisiae has also been demonstrated. So meaning, I think they have the same effect that it increase the stability of the plasmid. Okay? How about the protective environment? What do you mean by protective environment? So, but in the bacterial cells, the immobilization can provide protection to cells exposed to toxic or inhibitory substrates or environment. So that's one of the advantage of the immobilization because it provides a barrier to the cells that it will not heavily exposed to toxic like secondary metabolites that are already toxic. So that is why they will not be they will not be lysed, right? Because of the protected environment. So they can still be reused for the next production or the next fermentation. Okay, the immobilization of Cyclosporium sp and Pseudomonas potidas which have showed higher phenol degradation and tolerance. Great. So um, in this Trichosporium sp and Pseudomonas fotida, meaning they have it the phenol degradation and tolerance rates are increased, no? So because phenols are can be toxic to the the bac no, this is fungal cell. Pseudomonas, I think this is a bacteria. Ecosporium is a um, fungi, right? I think so. Yeah, and tolerance rate. Okay, so it really protects the toxic substances in the micro outside the environment. And moreover, studies show that alginate in trapped E. coli cells grown in trapped in calcium alginate showed low lipid to protein ratios even without the phenol in the growth medium. So, meaning showed low lipid to protein ratios even with that, meaning there is really an increase of cell growth here, even though ang phenol is a some supplemental medium for the growth of the E. coli cell. Okay, or maybe the lipid to protein in the yeah, yeah, that's it. Even though without the supplementation of the phenol in the growth medium. Okay. So another effect of immobilization of cells is the change of protein pattern of the outer membrane because there is an interaction between the proteins and the cell wall, right? So there are many different kinds of sorry, class. Different kinds of proteins that are attached to the cell wall. Either it is a positive, gram-positive bacteria or gram-negative bacteria. I mean, there is a change of the protein pattern. So, calcium alginate immobilized cultures of streptococcus cells were protected from the bacteriophage attack because of the exclusion of phage particles from the gel matrix. So, in other words, it really provides barrier protection that will protect them from cell death. Also, it was suggested that the immobilization of these bacteria could protect them from environmental stress, like the toxic compounds that are produced by the bacteria. So for the fungal cells, immobilization can create a protective environment. For the cells, as in the study in the co-immobilization of Saccharomyces cerevisiae cells with vegetable oils, cells could be protected against inhibitory substances. Okay? So these are inhibitory or toxic substances, secondary metabolites that are produced by the fungi because of a better solubility of the inhibitory compounds in the oil phase. Okay, another example is the increased tolerance of ethanol by immobilized brewer's yeast as reported by Norton et al. So I have already mentioned kanina that there is increased tolerance of ethanol, meaning you can really use the brewer's yeast because it is not susceptible anymore to the cell death in the higher concentration of ethanol. A decreased inhibition of ethanol productivity in entrapped Polyveromyces marcianus at high osmolality, according to Dale et al. 1994. So, it really provides protection and barrier talaga na it will not be susceptible anymore to cell lysis and cell death. So, I think that's the whole point there na it increased ethanol tolerance, it also increased osmolality tolerance, yeah, and ethanol concentration tolerance. How about the effects of mass transfer limitation? So in the bacterial cells, okay, the immunization can have an effect on the morphology, like of immobilized Lactobacillus helveticus, because of the delayed response time of entrapped cells to random pH changes. So meaning, there's a delay because of the thick barrier that the environment cannot easily access or have interaction between the cell wall and the fermenting the media. I mean, there is a delayed response that it could be that the bacteria will not uh, will not have the harsh, for example, the harsh pH or low pH, so it will not then respond like there is no susceptibility to cell death. Or it could be also that when the pH change, there is a slow response to the metabolism of the bacteria. According to Earth, the citrate metabolism, the Cocoon and device 1993, and lactate production of Lactobacillus lactis was altered because of the concentration and pH gradients in the gel heat. So, citrate metabolism and lactate production. Okay, next. So there is also another example. The germination time also of the Bacillus subtilis was significantly longer than for free cells. Okay, the germination or the budding effect after a time lag due to encapsulation. Yeah, the growth of the cells was uninhibited and no differences between entrapped and free cells were found. Okay. For the fungal cells, so in yeast growth cells, it was observed that there was a high invertase activity, okay, high inverse activity. So it converts it really degrades the sucrose into the to monosaccharides, okay, which was exhibited by immobilized cells due to a maintained expression of FUC2 gene and reduced susceptibility of the enzyme to endogenous proteolytic attack. So, because of the immobilization of the yeast growth cells, there is a high endogenous activity, and because there is also reduction to susceptibility of enzymes to the proteolytic attack from the outside environment. Okay, so these results have been interpreted in terms of diffusional limitations and changes in the pattern of invertase glycosylation due to the growth of yeast in an immobilized state. Another effect is that there is an enhancement productivity of enzymes and other products. Like for the bacterial cells, it has been observed that the production of several enzymes is increased upon immobilization. In the case of the study reported by Argyrakos about alpha amylase by alginate entrapped Bacillus amyloleucophaciens, enzyme production was decreased. So there is also reduction of the enzyme production for the Bacillus amyloleucophaciens, no, for the alpha amylase, no. So there is also a microorganisms or the bacteria that there is a higher there's higher production for other several enzymes. But for the Bacillus amyloleucophaciens, there is a reduction of the alpha amylase production. Okay, for the protease production by alginate entrapped Myxococcus santos cells was increased because of reduced inhibition by peptone and gelatin due to mass transfer limitation in the gel. For the fungal cells, a hydroxylase this is a hydroxylase enzyme from entrapped Mortariella isabelliana was found to retain its activity over longer period compared with free mycelia. Now, as a summary, the immobilization of microbial cells, specifically in bacterial and fungal cells, have different effects. As you have observed kanina, there is increased production, there is also a reduced production. That might be advantages or disadvantages of in the product formation of the cells. So you have to read a lot of articles if you are deciding to have an immobilization experiment. So and then you have to test the performance of the microorganisms in the particular in the kind of immobilization technique. Nevertheless, the application of immobilization of cells have been popular in terms of the production of enzymes and other important commercial products. Okay, that would be all, class. This is just so short, and I will now, do you have any questions, class? So if you have any questions about this topic, please don't hesitate to um, chat in our group chat, class. So please don't be shy. And then I will also give the journal article for this specific topic, a very detailed one, so that you can really read it. And I will now give you the learning task. Okay, so this is now the learning task part. So you have to answer these questions, and please remember that you have to write your learning task in a handwritten form. You have to take a picture of it and then convert to PDF and then submit it in the BSUE. So the number one question is, there are lots of advantages. No, I have already cited a few long. So, but then what could be the disadvantages of cell immobilization in the different culture fermentation? And then second is that research one journal article published in the year 2015 to 2020 about wine fermentation that uses immobilized cells. Now, summarize the main objective. So it should not be long class, ha? It should not be that quite very long because I will be having hard time to read it. But then a one page will do, just the direct to the point, the main objective, the methods used. You can really don't copy the very details of the methods, but it's just the framework or the summary of the methods used, then the results and discussion, and the conclusion and recommendation. Now, don't forget to cite the references. So, reading journal articles class are really vital in the research academe, in the academe, and also it can really widen your perspective and in the knowledge that you are right now, especially in biotechnology and this course, the bioprocess one. So that's it for the learning task class. Do you have any questions about it? So again, if you have a clarification, so please chat on the group chat for our for clarification. I'll be willing to reply that. No, so but there are many times that there are delayed response that you have encountered from me, especially when you chatted beyond 5 p.m. So, and also if you happen to chat me on a weekend, so I have that choice not to reply. But then during weekdays, I'm really available to reply to answer your queries. Okay, so I think this is the last slide class for the cell immunization technique. So I will then send also the we will next week we'll now proceed to module 4. Okay, so malapit na tayong makatapos class for this sem. It's December next week, so Merry Christmas and a Happy New Year class. So I hope that you are enjoying your sem, no? So and I hope that magkikita-kita na tayo next sem. We will have a limited face-to-face class. Okay, that's all. Okay, thank you so much class for watching this video, and God bless all of you. Bye-bye.